Alright guys, welcome back to Underground Science, and in this video we're going to be talking about base excision repair, okay? So remember we talked about non-homologous end joining and its relationship to specifically ataxia, telangiectasia, and then we talked about homologous recombination and its relationship to Fanconi anemia. Now those two were double-stranded repair mechanisms. This base excision repair, right, is going to be a single-stranded repair mechanism. Alright, so let's go and get started and see how this works. So we have our template strand and coding strand, right? So let's say our top strand is, remember DNA is arranged in anti-parallel fashion. So 3' to 5', then the bottom strands from 5' to 3'. This X represents the wrong base, because remember bases are supposed to be complement to one another. So for example, in DNA, adenine will bind to thymine, and cytosine will bind to guanine. If it was RNA, adenine would bind to uracil because in RNA you don't have thymine, okay? So here, this X represents something that's not guanine, okay? Because we're supposed to have guanine here because this is cytosine here. So we got to fix this, okay? So the first step in base excision repair, right, is a base-specific glycosylase will come in. So if we see ACE here, we know there's an enzyme, right? So base-specific glycosylase will come in. So it's specific to the wrong base, okay? So it's going to come here, and it's going to detect that, oh, this is not the right base, meaning it's not guanine, because this is cytosine, so this has to be guanine, right? So it's going to detect, okay, this is not guanine, so let's cut this out. But notice it only cuts the base out, and the sugar phosphate backbone in blue is mostly intact. So when the base-specific glycosylase cuts the wrong base out, what happens is it leaves you with an AP site, okay? So now that site stands for an apurinic site or an apyrimidinic site, okay? So an apurinic site or an apyrimidinic site. That's an AP site for short. And the reason why is like, if this were an adenine or a guanine, which are the purines, and you cut that out, that leaves you with an apurinic site. But if you cut out for example, like a thymine, which is a pyrimidine, that leaves you with an apyrimidinic site, okay? So that's why it just leaves you with an AP site, okay? So we cut the wrong, so like in this case, right, let's say this wasn't guanine, right? And it was, let's say, I don't know, like thymine, right? And we cut this out, then that would be an apyrimidinic site because we just removed a pyrimidine, okay, which thymine's a pyrimidine. Okay, so we make an AP site after base-specific glycosylase comes in and cuts the base out, okay, the base we need to replace. Then what happens is an AP endonuclease comes in, okay? So this endonuclease is cool because what happens is this AP endonuclease comes in, and you see, remember, the blue represents the sugar phosphate backbone, which is still intact. We still need to cut that out because right now we only cut the base out. We need to cut the whole nucleotide out. Okay, so that's base plus sugar phosphate backbone. So AP endonuclease is going to come in and go to the 5' site. Okay, so notice how this is 5' over here. So this side is the 5', this side is the 3'. Okay, see, 3'. So AP endonuclease come in and cleave the 5' end of the sugar phosphate. And then AP lyase is going to come in and cleave the 3' end of the sugar phosphate. Then that's going to lead to the sugar phosphate is going to be taken out, right? Then afterwards, what can happen is you can have DNA polymerase beta that will come in and put the correct nucleotide in this case, right? So this is cytosine, so then we have to have a guanine here. So DNA polymerase beta will come in, put a guanine, which is the correct base, right? And then DNA ligase can come and seal any sort of like gaps after that. Okay, so that's literally base excision repair. One thing to know is like, when does this happen? So this basically happens when sometimes a sudden deamination occurs. So you lose an amine group from a base and then it changes to the wrong base. Okay, so if we're talking about deamination, we should know that if you take cytosine and you deaminate it, it can actually turn into uracil. If you take guanine and deaminate it, it can turn into xanthine. And if you take adenine and deaminate it, it can turn into hypoxanthine. Okay, so these are all deamination reactions. Alkylation is another thing where you can stimulate base excision repair. If you see alkylation of the bases of your DNA, right? So alkylation. All right, so the main things to know is deamination and alkylation of your DNA can lead to base excision repair. Okay, so that's basically it for base excision repair. And we're going to be talking about more types of repair in our future videos. So thanks for watching, and I hope to see you guys in the future.